Hello, am I audible? Good evening to the students who have joined. Okay, thank you so much. Okay, first. Please let me know if the PowerPoint presentation is visible. Yeah, very good evening. Okay, thank you so much. We will wait till it's 6 p.m. and more students join, and then we will start today's session. Meanwhile, let me know if there is any problem with the audio or the presentation. So I will wait for a few more minutes. Maybe I will wait for 6 p.m. and then I will start the session. The student who have already joined, please wait till at 6 p.m. Then I will start the session. Okay, so very good evening. I hope my voice is audible and the screen was visible. So let's start with the introductions. Meanwhile, I hope more students will join eventually. Please let me know if there is any problem with the audio or the presentation. Otherwise, I am going to start the session. So very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development, and today it is our week 5 live session. So every week we a particular week or particular portion of the course is available to you, and right now the week 6 course contents is available are available on the SWAYAM portal, and today we will discuss the topic few questions regarding to the week 6 course content. Since we are going ahead with every week, like for the first live session we covered week 1 and week 2 course content all together, and subsequently today it is our week 5 live session where we are going to cover up the week 6 course content. So before starting, I would like to say a few things that I repeat before every class. So there will be this kind of problem solving session every week, and the sessions will be on every Monday from 6 p.m. to 8 p.m. The sessions will be live on Google Meet platform. The link is same, and the link is shared to you before, I think, 1 day before of the class. Okay. So if any network disruption happens during the class, please do not leave the session. Okay, I will try to join as soon as possible, plus please do not leave the session since the weather is not very good today, and so if there might be any kind of network problem, so if there is any network problem, please let me know in the chat box. I will try to rejoin, and if there is any network issue from your side, you can rejoin it at any time. I have made the call open for all, so you can rejoin at any time if there is any network issue from your side. So during the session, I have I am recording the today's Google Meet session. Okay, so for the smooth recording, I have muted your microphones and also the videos so that there is no disruption for recording. But the chat box is always open. I am checking the chat box every now and then. So if you have any doubt, any query, or you want to tell me anything, you can communicate through the chat box. If still the doubt persists after the class also, you can let me know via email. I will share my email ID with you, okay, at the end of the session, and you can let me know via email also that if there is any problem, and this presentation and class recording, it will be shared to you. Okay, after the class I will try to post it as soon as possible, the the presentation and also the class recording. So I hope everything is fine. Then I am going to start today's session. Let me check. Okay, so the chat box is always open. So like we do every class, I will show you a question. Okay, today we are going to discuss the week 6 course content. So I have given in the presentation, I have collected 20 questions related to the week's course content. All these questions are from week 6 course topic. Okay, the week 6 is right now live, so these questions will help you to understand the week 6 course content better, and also it will help you to answer your weekly assignments. Okay, the weekly assignments, you know, these are presented in the SWAYAM portal. If you go to the SWAYAM portal, you will find it here. If you log into the SWAYAM portal, you will find the weekly assignments here, and here you will find the weekly quiz, and also you will find the problem solving session here. By clicking it here, you will be directed to a page where all the presentations or the links and all the videos are uploaded. Okay, so you can check it out after the class. So let us start with the week's content. So today it is our week 5 live session, and we are going to cover the week 6 course content of organic chemistry in biology and drug development. So what is in the week 6? So week 6 is basically we will start with the synthesis of oligonucleotide, and we will start with the central dogma. So central dogma is a very important topic which comprises of the DNA replication, transcription and translation, and protein synthesis also. So we will discuss things related to the synthesis of oligonucleotide and the enzymes which are important for DNA replication, transcription, translations and overall things. Okay, so let us start with the first question. So the first question of today's session is, in the cell, protein synthesis takes place in, you have to fill in the blanks. The correct answers, the options are given as like A, mitochondria, number B, nucleus, number C, Golgi vesicles, and number D, ribosome. So you have to identify in the cell where the protein synthesis takes place. Is it mitochondria, is it nucleus, is it Golgi vesicle, or is it ribosomes? After showing you a problem, I will wait for 2 to 3 minutes. Okay, all of you try to answer the question in the chat box. Let me know A, B, C or D, which of the following options you think is the correct one, and then I will show you the correct answer and we will discuss it. Okay, since there is no marks on answering the questions in the class, so I want all of you to participate. Okay, even if your answer is wrong, it does not matter, but participation is very important. So let me wait for 2 to 3 minutes, and then I will check the chat box, and then I will show you the correct answer. So the first question is, in the cell the protein synthesis takes place where? Where the protein synthesis takes place in the cell? Mitochondria, nucleus, Golgi vesicle or ribosomes? Okay, let me check the chat box. Okay, I have few answers. It says number C, number D. Okay, I have two different options for problem number 1. So let us check if it is number C or is it number D. Which of the following is the correct answer for problem number 1? Okay, so the correct answer of problem number 1 is, yes, it is number D, ribosome. So in the cell, protein synthesis takes place in ribosome. So basically, what are ribosomes? So ribosomes are small, rounded and dense particles of ribonucleoprotein. They occur either freely in the cytoplasm and the matrix of the mitochondria and chloroplast, or remain attached with the membranes of the endoplasmic reticulum and nucleus. They are the size of the protein synthesis in both prokaryotes and eukaryotic cells. the cells in which active protein synthesis takes place, for example pancreatic cells, hepatic parenchymal cells, plasma cells, thyroid cells, and the ribosomes remain attached with the membranes of RDR. Okay, and then cells which synthesize the specific proteins for the intracellular utilization or storage, like erythroblasts, skin, they etc. contain large number of free ribosomes in the cytoplasm. Ribosomes are usually designated according to their rates of sedimentation. The sedimentation coefficient is expressed in the Sedgwick unit or S unit. This S is related with the size and the molecular weight of the ribosomal particle. So, there are two type of ribosomes basically. So, first one is the prokaryotic ribosome and the second one is eukaryotic ribosome. So, if the prokaryotic ribosome, it has two different subunit. It has 150S subunit and the small which is the larger subunit, and one smaller subunit which is the 30S subunit, and in total it is said to be 70S subunits. Okay, and eukaryotic ribosome is total is the 80S subunit, and the larger subunit it has 60S subunits, and the smaller one has a 40S subunit. In the largest 50S subunit of the prokaryotic ribosome, it has 5S RNA and 23S RNA, where in the smaller one has 16S RNA, and for the bigger subunit of the eukaryotic ribosome, it has 28S RNA and with the 5S RNA, and then it has a 40S in the 40S subunit it has the 18S RNA. Okay. So, remember the protein synthesis in the cell happens in the ribosomes, and that there are the different ribosomes for prokaryotes and eukaryotes, and the difference are in the size and their units, and the units are in the Sedgwick subunit, which is related with their size, molecular weight of the ribosome particle. Okay. So, the correct answer was number D, and in the chat box there were number D. So, very good. Now, I will quickly move toward the next problem. So, problem number 2 says, what role does messenger RNA play in the synthesis of proteins? Messenger RNA means mRNA. So, you have to identify the correct role of mRNA in the protein synthesis. Your options are: number A, it catalyzes the process; number B, it provides the genetic blueprint for the protein; number C, it translates the genetic code to a specific amino acid; and number D, it modifies the messenger RNA molecules prior to the protein synthesis. So, what does the messenger RNA or mRNA do in protein synthesis? What is its prime role? Again, I will give you 2 to 3 minute time. Read all the options carefully and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 2. All of the present students, I want all of you to participate. Even if your answer is wrong, it's just fine, but try to answer the question. What the role does the messenger RNA or mRNA play in the synthesis of protein? It catalyzes the process, it provides the genetic blueprint of the protein, it translates the genetic code to a specific amino acid, it modifies the mRNA molecules prior to protein synthesis. Okay, let me check the chat box now. Okay, I have few answers, like number C is the most given answer. Okay, let me check if number C is the correct one or not. So the correct answer is number B. It provides the genetic blueprint of the protein. So basically, what is the mRNA does? So mRNA is the starting point of the translation. So the DNA is transcribed into the mRNA. Okay, mRNA translates into the amino acids, and each codon correspond to a particular amino acid. The amino acids form a polypeptide chain, and the polypeptide chain folds into the protein. Okay, so as I can see here, the number C option is also correct here. Okay, so the both B and C are the correct answers for this question. I will just update the answer later. Okay, so both the B and C are the correct answer for problem number 2. Okay, it provides the genetic blueprint for the protein, and how it does it? Because it translates the genetic code for a specific amino acid. The mRNAs translates the genetic code. This answer is also correct. I will update it later. Okay, so both B and C are the correct one. Now let us check how this particular protein synthesis happens and how DNA directs this kind of protein synthesis. So first we have discussed about the messenger or mRNA, which is a starting point of the translation. Okay, now how the DNA directs the protein synthesis. So basically we have this double stranded DNA in the cell nucleus. Okay, and as you can see, there are alternating sugar and phosphate groups here, and the strands of DNA, first the strands of DNA, they unzip and allow free RNA nucleotides to link the separated strands. Okay, this is a messenger RNA forming on the DNA strand. As you can see here, messenger RNA or mRNA forming on the DNA strand. Here it is mentioned. The free RNA nucleotide approaches the unzipped DNA molecule to pair its base with the DNA nucleotide. From here we have the mRNA that moves in the cell nucleus to a ribosome. This is the ribosome molecule which I have shown you in the last slide. So this is a mRNA moves from the cell nucleus to ribosome in the cytoplasm. There it acts as a pattern on which amino acid transported by the transfer RNA or tRNA from the protein. The ribosome moves left to right along the mRNA. Okay, and then we have the formation of protein on the ribosome. So how it happens? So there we have a tRNA. This tRNA have a attached amino acid, and there is an anticodon sequence here which matches with the codon sequence on particular mRNA, and then it brings the amino acids, and the amino acids are joined to all together with a peptide bondish bond, and the growing polypeptide chain eventually constitutes a protein. And this process takes place until and unless there is a stop codon or a particular set of nucleotide, particular sets of the base, okay, which will guide the particular protein translation step to stop, and then the growing polypeptide chain is detached from the tRNA, and it is ready for the next step. So that's how the particular protein synthesis takes place. I have just given you an overview of it. We will discuss it later on also in the detail. So here I am just going to correct it right now, otherwise I might forget later. So here the question, the option number B and C both are correct. Okay, for mRNA it acts as a blueprint for the genetic blueprint for the protein synthesis, and also it translates the genetic code for a particular amino acid. Okay, so yeah, both B and C was the correct answer. So now I will move toward the next problem. So problem number 3 says, which of the following is a start codon and for which amino acid it codes for? Okay, so I have discussed a little bit about the codon here in the last slide. So you have to identify now which of the following is a start codon and for which amino acid it codes for. Your options are number A, AUG and methionine, number B, UCU and serine, number C, UUU and phenylalanine, and number D, AAA and lysine. So which of the following is the start codon and for which amino acid is coded for? So basically in the mRNA there are three nucleotide bases. I mean the adenine, guanine, thymine, cytosine or uracil. Okay, not a thymine, uracil. So there are combination of this particular bases in the in a collection of 3, and this is known as the codon. A particular codon codes for a particular amino acid, means a particular codon, it gives us a blueprint about a particular amino acid. Wherever there is a codon is present, that particular amino acid will add to a particular in a growing polypeptide chain. So now you have to identify which of the following is the start codon and the amino acid for which it codes for. So the start codon is always same for any kind of polypeptide chain or protein synthesis. Okay, it will be same. So you have to now identify which of the following options are correct, A, B, C or D. I will wait for some time and then I will check the chat box. Which of the following is the start codon and for which amino acid it codes for? Okay, let me check the chat box. Okay, I have one answer which says it is number A. Okay, let me check if number A is the correct one or not. Okay, so the correct answer is yes, it is number A, AUG and methionine. So the start codon is AUG and methionine is the amino acid for which it codes for. So transcription, basically it is the first step in the protein synthesis, formation of mRNA. The transcription means it is basically the formation of the mRNA in the nucleus by the complementary base pairing with the DNA. Okay, so the first step of the protein synthesis is transcription, which is the formation of mRNA in the nucleus by the complementary base pairing with DNA. mRNA then leaves the nucleus and travels to the ribosome. Ribosome read the mRNA code, and codon, it is a sequence of 3 bases of mRNA. So there are 64 different codons and 20 different amino acids, and several amino acids have several different codons that the code for them. One codon is known as start codon, which is AUG, and it codes for the methionine. Okay, so remember that in the protein synthesis the first step is the transcription, and this which is the formation of mRNA from the nucleus by the base pair, complementary base pairing of DNA. Then the mRNA leaves the nucleus and travels to the ribosome where the protein synthesis takes place. So what happens in ribosome? In ribosome, the ribosomes read the mRNA code, and the codon is the sequence of 3 bases, a particular combination of 3 bases. So there are 64 different codons. So we know there are 4 different bases in RNA like adenine, guanine, uracil and cytosine, and combination of this 4 gives us 64 different codons, and there are 20 different amino acids. So several codons, they code for a particular same amino acid. It can happen. So one codon is known as the start codon, which is AUG, and it codes for the methionine amino acid. Okay, so I hope it is clear. So if you have any problem, any doubt, let me know. So answer number A was the correct one for the question number 3. Okay, AUG and methionine is the correct answer. So now I will go to the next problem. Problem number 4 says correct order of the steps of the solid phase oligonucleotide synthesis is, your options are number A, deprotection of DMT group, coupling and deprotection of base. Number B, coupling, oxidation of phosphorus, deprotection of DMT group. Number C is deprotection of DMT group, coupling, oxidation of phosphorus. And number D, deprotection of DMT group, oxidation of phosphorus and coupling. You have to identify the correct order of the steps for the solid phase oligonucleotide synthesis. Again I will repeat the options. Number A, deprotection of DMT group, coupling and then deprotection of base. Number B, coupling, oxidation of phosphorus and deprotection of DMT group. Number C is the deprotection of DMT group, coupling followed by oxidation of phosphorus. And number D is the deprotection of DMT group, oxidation of phosphorus and coupling. Which of the following is the correct order of steps of the solid phase oligonucleotide synthesis? Read all the options carefully, think how the solid phase oligonucleotide synthesis takes place and then answer the question. Correct order of steps of solid phase oligonucleotide synthesis is, identify the correct option. Number A, deprotection of DMT group, coupling, deprotection of base. Number B, coupling, oxidation of phosphorus, deprotection of DMT group. Number C, deprotection of DMT group, coupling and oxidation of phosphorus. And number D, deprotection of DMT group, oxidation of phosphorus and followed by coupling. Which is the correct order? Okay, let me check the chat box now. Okay, I have one answer which says it is number D. Okay, let me check if number D is the correct answer or not. Others also please try to answer the questions. Even if your answer is wrong, it does not matter, but participation is very important. Okay, so for problem number 4, the correct I have got one answer in only in the chat box which says it is number D. Now let me check if number D is the correct one or not. So the correct answer is, okay, it is the number C, the deprotection of DMT group, coupling and the oxidation of phosphorus. It is the correct order of the solid phase oligonucleotide synthesis. This is a cycle of the solid state oligonucleotide synthesis. So as you can see, the cycle starts from here. So we have a solid support, okay, the support is in bed which is acts as a solid support. It is very similar to the solid phased peptide synthesis which we have discussed earlier, where the mediphyl resin is the solid support. Here also we have a resin support, and then we have a particular nucleotide which has this base which denoted as base n minus 1, and we have a 5 prime hydroxyl group which is protected with a DMT protection group. Okay. So, the first step is the deprotection. Okay. This DMT group is first, it has to be deprotected. Acid catalyzed removal of DMT allows the subsequent base addition. Now, again we have another nucleotide. Okay, and this nucleotide has this is the base is denoted as SN, and this also has the DMT group protected in the 5 prime region, and we have in the 3 prime position there is a phosphorus. Okay, there is a phosphorus group which is acts as a protecting group. So, now what happens in the second step is the coupling step. A DMT protected phosphorylamide is added to the unprotected 5 prime using a tetrazole activator. Okay. So, here the this for base N we have the 3 prime position, and then we have in this DMT protected group. So, the unprotected the oligonucleotide which is unprotected is added to this the second oligonucleotide, and the coupling takes place using a tetrazole activator. And then the third and fourth step is the capping step, where the unreacted 5 prime OH is acylated to prevent further chain extension, and then the oxidation of phosphorylate ester to phosphate using the aqueous iodine it takes place. Okay. So, oxidation of phosphorate is the third step which takes place using the aqueous iodine, and now it is this particular oligonucleotide it is released, and the cycle it starts from the next step. Okay. So, that is how this particular solid phase nucleotide oligonucleotide synthesis happens. So, the first step is the deprotection of the DMT group, then followed by the coupling, and then the oxidation of phosphorus. Okay. So, the option C was the correct one. Now, I will move toward the problem number next problem. So, problem number 5 says which of the following statements is true regarding the solid state DNA synthesis. Your options are: number A, starting point is 5 prime OH and it is attached with the silica. Number B, 3 prime OH is protected as DMT group. Number C, T does not need to be protected. T means thymine. And number D, G means guanine is protected as benzoyl. So, which of the following statements is true regarding the solid state DNA synthesis? Again I will repeat: number A, starting point is 5 prime OH and it is attached with silica. Number B, 3 prime 3 OH is protected as DMT group. Number C, T does not need any protection, and number D, G is protected as benzoyl. So, which of the following statement is true regarding solid state DNA synthesis? You have to identify. I will wait for 2 minutes and then I will check the chat box. So, which of the following statement is true regarding solid state DNA synthesis? Number A, starting point is 5 prime OH and it is attached with silica. Number B, 3 prime OH is protected as DMT group. Number C, T does not need to be protected, and number D, G is protected as benzoyl. You have to identify the true statement. So, regard this kind of true false kind of question is sometimes tricky. So, you have to read the question and all the options very carefully to answer these questions correctly. let me check the chat box now. Let me see if there are any answers for this problem. Okay, I have few answers. It says number B. Okay, number B is said to be the correct answer. Let me check if number B is the correct one or not. So, the correct answer is the statement which is true regarding solid state DNA synthesis is, it is number C, which says that thymine does not need to be protected. So, as I have mentioned in the last slide itself, that the starting point is not the 5' OH. Okay, as you can check the last slide, as you can see, the 3' OH is attached with the silica. This is the third position, right? This is the 3'. Okay, let me use the laser pointer. So, this is the 3' position which is attached with the silica, and 5' OH, it is protected as DMT group. Okay, so the first two statements are false regarding that. So, thymine does not need to be protected and guanine is protected as benzoyl. So, there are two statements which we have to check. So, the statement number C is said to be the correct one. Now, let me show you. So, these are the structures of adenine, guanine, thymine and cytosine, right. So, if you look at the structures very carefully, you will find there is a striking difference between adenine, guanine, cytosine and thymine. What is the difference? The difference is like the adenine, guanine and cytosine, they have a primary amine group or NH2 group which is free, whereas in thymine we do not have any free NH2 group. So, that is why thymine does not need any protection, since it does not have any free NH2 group. So, why this kind of protection need to be happen? Okay. So, this is the initial, this is the structure which I have not shown in the last slide. So, this is the CPG bit and this is the solid state support. Then we have a spacer molecule which is a long chain polymer type of molecule, followed by the initial nucleotide. So, the initial nucleotide has a base in the first position and followed by the third position OH which is attached with the silica or the solid state, and then the 5' OH which has the DMT protection group. Okay. So, the base there can be any adenine, guanine, thymine or cytosine, any kind of base, it can be present in this position. The amino groups of adenine, guanine and cytosine are derivatized by the addition of benzoyl, isobutyryl and benzoyl groups respectively. So, adenine is protected as benzoyl, guanine is protected as isobutyryl and cytosine is protected as benzoyl. Okay. Thymine does not need any protection because it does not have any free NH2 group. So, why do we need to protect the other bases, I mean adenine, guanine and cytosine? Why do we need to derivatize them? Because their free NH2 group can hamper our solid state oligonucleotide synthesis in this phosphorylamidate method because of the there are chances of side reactions due to their free NH2 group. Since the thymine does not have any amino group, so there is no chance of this particular side reaction, so it does not need any protection. Then we have a spacer molecule which is attached to the solid support controlled by the pore glass. Initial nucleoside, the base plus sugar is attached with the spacer by the 3' terminus, and the initial nucleoside has been protected by 5' DMT or dimethoxytritryl group. Washing with the acetonitrile to remove water and any nucleoside, and flushing with argon to purge the acetonitrile. So, this is the steps of the this is the initial steps of the solid state oligonucleotide synthesis. So, remember there are 5' position, it is the DMT protection, there is which is the dimethoxytritryl which can be removed with the aqueous acid. Then we have this 3 position which is combined with the spacer molecule and joined to the solid support. The bases, the adenine, guanine, cytosines are protected with benzoyl, isobutyryl and benzoyl group respectively, and thymine does not have any NH2 group, so it does not need any protection. Okay. Now, I will move toward the next problem. So, problem number 6 it says that which of the following statements regarding DNA synthesis is true? Your options are: number A, enzymatically it takes place from 3' to 5' direction only. Number B, enzymatically it takes place from 5' to 3' direction only. Number C, chemically it takes place from 3' to 5' direction only. And number, okay, I have I think number C and number D, both options are same, so I will just correct it. And chemically it takes place from 5' to 3' position only, let us say. Okay. So, it is 5' to 3'. I will correct it later on. So, take it as like this. So, your first option is enzymatically it takes place from 3' to 5'. Enzymatically it takes place from 5 prime to 3 prime, chemically it takes place from 3 prime to 5 prime direction, and chemically it takes place from 5 prime to 3 prime direction only. These are the 4 statements from which you have to identify that which statement is true regarding the DNA synthesis. Which of the following statements regarding DNA synthesis is true? Okay, let me check the chat box now. Okay, I have answer number A and then I have number C. I think it is for, yeah. So number C and number A, I have two different answers. Okay, let me check which of the following is the correct answer, number A or number C. So the correct answer is, it is number B. It takes place enzymatically from 5 prime to 3 prime direction only. This statement is true. Okay, first let me correct the things. Okay, there will be, this will be 5 prime and this will be 3 prime. Okay, the options were, two options were same, so I have changed it. So enzymatically it takes place from 5 prime to 3 prime direction only. This statement is true regarding the DNA synthesis. Okay, so DNA replication synthesis of new DNA strands. So DNA polymerase is the enzyme which basically is main in this case of DNA replication. So DNA polymerase with the RNA primer in place, DNA polymerase enzyme catalyze the synthesized new DNA strand in the 5' to 3' direction. Okay. So there is a RNA primer and DNA polymerase, it joins the nucleotides to form a new new DNA strand from 5' to 3' direction only. So DNA polymerases are the enzyme that synthesize the DNA molecule from deoxyribonucleotides, the building blocks of DNA. These enzymes are essential. So DNA polymerases is not a single enzyme. Okay, so there is a class of a group of DNA polymerases with the different functions, but their main thing is that they synthesize help in the replication and synthesize the particular new DNA strand. So these enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from single original DNA molecule. So during the process, DNA polymerase reads the existing DNA strands to create two new strands that match the existing ones and also performs the proofreading and error correction. So DNA replication, there are some properties regarding the DNA replication. So DNA replication is semi-conservative. Each new DNA strand serves as a template for the synthesis of the new strand, producing two new DNA molecules, each with the one strand new and one strand old. This is called the semi-conservative replication, where in between two strands there are one is new and one is old one. Replication begins at an origin and usually proceed bidirectionally. The replication loops always initiate at a unique point which was termed an origin. A and T base pairs in the rich sequences. DNA synthesis proceeds in a 5' to 3' direction and is semi-discontinuous. A new strand of DNA is always synthesized from 5' to 3' direction with a free 3' OH at the point at which the new DNA is elongated. Okay. So statement number B was the correct statement regarding the DNA synthesis. So now I will move toward the next problem. Problem number 7 says, which of the following is a stop codon? Your options are number A 5' AUG 3', number B 3' AAU 5', number C 3' UAA 5', and number D 5' GCC 3'. You have to identify which of the following is a stop codon. Look all the options very carefully and then answer this question. Your option A is 5' AUG 3', number B 3' AAU 5', number C 5' UAA 3', and number D 5' GCC 3'. Again I will wait for 2 minutes and then I will check the chat box. Which of the following is a stop codon? Let me check the chat box now. Okay, so I have one answer, which is, it is number C. Okay, let me check if number C is the correct one or not. So the correct answer is, it is number B, 3' AAU 5' is the stop codon. So that's why I have told you to read the options very carefully. So basically the codons, how we read the codons is from the 5' to 3' end. Now if you look at the options very carefully, so this is the particular way of a codon. This 5' AUG 3', it is a start codon and it codes for the amino acid, which is the methionine. Now if this UAA is the stop codon indeed, but you have to check that there is a 3' and 5'. So 3' UUA, basically it reads as 5' AAU. Okay, so it is not a stop codon. This stop codon is, the correct answer was number B. Okay, because it reads as the 5' UAA and 3'. So UAA is a stop codon, but you have to check also the sequence of the sequence of the codons. Okay, so 5' UAA 3' is the correct order. It will start from 5' and end with the 3'. So that's why number C is not correct, because it is saying 3'. So basically it reads as 5' AAU 3', okay, which is not a stop codon. Now let me show you. So those codons, they provide the signal for the termination of the polypeptide chain, are known as the stop codons or termination codons. So there are 3 termination codons, UAA, UAG and UGA. Since stop codon signals do not signal codons do not code for any amino acid, they are earlier called the nonsense codon also. So as you can see, there are positions, and based on that we have this. So you have to remember this order is from 5' to 3'. Okay. So UUC, so these are for phenylalanine, these codons are for leucine, these codon are also for leucine, these are for isoleucine. This is AUG for the this is for your start codon for methionine. Then these codons are for valine, followed by this. So UAA, it is the stop codon, UAG is the stop codon, and UGA also is the stop codon. But you have to remember the order also, it should have to, it will start from 5' and end with the 3'. So since this codon, which you said is the correct one, the number C, which is 3' UAA 5', it is basically reads as 5' AAU. So you can check AAU, is there any codon for AAU? Yeah, AAU, it codes for the asparagine residue. Okay, so it is not a stop codon. So correct answer was number B. So is it clear? I know this kind of problem is sometimes it is difficult to understand, because this there are many students make mistake in this kind of problems. So you have to be careful whenever there is this type of question. Okay. So now I will move toward the next problem. So the problem number 8, it says that the position where the tRNA will bind to the ribosome depends on its number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, and number D, hairpin like structure. So the position where the tRNA will bind with the ribosome depends on it which kind of sequence. Is it codon sequence, is it intramolecular hydrogen bonding, is it anticodon sequence, or is it hairpin like structure? Where tRNA will bind with the ribosome. The position where tRNA will bind with the ribosome depends on its number A, codon sequence, number B, intramolecular hydrogen bonding, number C, anticodon sequence, and number D, hairpin like structure. So the position where tRNA will bind the ribosome depends on it. Let me check the chat box. Okay, I have two answers. One says that it is number C and another says it is the number A. Okay, let me check which of the following is the correct one. So the correct answer is, so the position where tRNA will bind with the ribosome depends on it. Yes, number C is the correct one, anticodon sequence. Okay, codon is for the mRNA. You have to remember codon is for the mRNA, anticodon is for the tRNA. Now let me show you. So this is the tRNA. So we have a mRNA which have a 5 prime to 3 prime, and we have a particular codon sequence here which reads from 5 prime to 3 prime, as I have mentioned in the last slide. Then we have a tRNA. So this is the structure of tRNA, where there is a 3 prime position where the amino acid binds. Okay, and this amino acid will bind based on the anticodon sequence. So anticodon sequence on the tRNA is a complementary binding mode for the codon sequence. Okay, then we have, this is the shape of the tRNA which looks like a T. So we have a T-Chi-C arm, there is a variable arm, we have an anticodon arm, and then we have a D arm or acceptor arm. Okay, so remember the anticodon is the sequence where it will bind to the mRNA which is the complementary to the codon sequence of particular amino acids. So now let us see how the particular translation or protein synthesis takes place. I will give an overview of it. So we have the mRNA molecule which is from 5 prime to 3 prime, and there are codons. Okay, there is a AUG which is a start codon, and we have different stop codons like UAG, UG and UAA. Then we have a ribosome, a large subunit and small subunit of ribosome, and tRNA, it comes with the first amino acid. So AUG is the start codon. So what is the anticodon for the start codon, which is the which codes for the amino acid methionine? It is the UAC, which is complementary to the AUG. So now first is the initiation step. So what is the initiation step? So the translational complex forms, and tRNA brings the first amino acid in the polypeptide chain to bind to start the codon for the on the mRNA. Okay, so first the translational complex is forming with the large subunit, small subunit of the ribosome with mRNA and tRNA, and there the first amino acid is brought up by the tRNA, and it will start the protein synthesis. Then the second step is the elongation step. tRNAs, different tRNAs will bring the amino acid one by one based on their anticodon sequence, which is again will be for the based on the codon sequence, and the elongation of the particular polypeptide chain will happen, followed by the termination, which is release factor recognizes the stop codon. Okay, so there will be a stop codon, as you can already, I have mentioned that UAG, UG or UAA is the stop codon. So there will be stop codon also at any position of the mRNA, and there will be a release factor. This release factor recognizes the stop codon, translation complex dissociated, and completed polypeptide chain is released. Then the cycle or the components of the translational complex, the ribosomes, the tRNA, mRNA, all are the recycled, and then new cycle starts. Okay, so remember anticodon sequence is the position where the tRNA will bind with the ribosome. So I think it is clear. Is there any doubt? If there is any doubt, let me know. Okay, at any point of any class, at any question, if you have any doubt, let me know then and there in the chat box. I will repeat that particular question if you have any doubt. Okay, so now I will move toward the next problem. So problem number 9, it says what would be the complementary RNA strand for the following coding DNA sequence in the transcription process. So what will be the complementary RNA strand for the following coding DNA sequence in the transcription process? The DNA sequence is given as 5 prime GTTA CGTG C3 prime. You have to identify the complementary RNA strand in the following coding DNA sequence on the transcription process. Your options are number A 5 prime GUUACGUGC3 prime, number B 3 prime CAAUGC ACG 5 prime, number C 5 prime CAA UGC ACG 3 prime, and number D 3 prime GUU ACG UGC 5 prime. So what would be the complementary RNA strand for the following coding DNA sequence in transcription process, which reads as 5 prime GTTA CGTGC 3 prime. Again I will wait for some time, then I will check the chat box and we will discuss it. Okay, let me check the chat box now. Okay, I have one answer, it says number D. Okay, let me check if number D is the correct one or not. So the correct answer is, which is number A, 5 prime GUU ACGU GC 3 prime is the correct answer. Now let me show you how to do this kind of question. So basically first we have the two strands, from one DNA strand is from 5 prime to 3 prime and another is from 3 prime to 5 prime. Okay, so the sequence which is given here is 5 prime GTT ACG TGU 3 prime. So the question was the complementary RNA strand which you have to identify, the complementary RNA strand for the following coding DNA sequence. Okay, so there are two strands in a particular DNA. One goes for 5 prime to 3 prime, which is known as the non-template strand or the coding strand or the sense strand, and another one is the template strand or the non-coding strand or the anti-sense strand. Okay, so here it is said that the complementary RNA coding DNA sequence is given. Okay, so what is the coding DNA sequence? The coding DNA sequence is like this. The coding DNA sequence is 5 prime GTT AUGCG TCG 3 prime, and you have to identify the complementary which is your particular complementary RNA strand. So as you can see the coding sequence, so basically whenever the question is asked like this, that you have to identify the complementary RNA strand. So remember the RNA strand, first you have the RNA strand will go from the 5 prime to 3 prime. Okay, so RNA strand will the template strand is and the non-template strand, there are some difference. So non-template, the template strand is for where the RNA will be synthesized. Okay, so here the template strand is this one, and the 3 prime to 5 prime, and the RNA is the complementary of the template strand. Okay, so as you can see that RNA for the complementary template strand is the same with the your non-template or coding strand, but the difference is that there will be the only the position of T will we have U. So how to solve this kind of question, let me now show you. So this is the strand for the, you have to remember this thing, coding strand. If it is the coding strand, so what will be the non-coding or template strand? It will be just opposite to it, right? So it will be your 3 prime. For G we will have C, for T we will have A, we will have C, A, then for A we will have your T, then for C we will have G, for G you will have C, for T we will have A, then for G we will have C, and for C we will have G, and we will have it as a 5 prime. So it is of the your non-coding. So if it is the non-coding means it is for the template strand. Now the RNA, it will be complementary of the template strand. So if you do the complementary for this template strand, which is the for RNA, so what it will give us? It will give us the 3 prime will be become 5 prime. Now the C, it will become G. For A, the complementary, in case of RNA it will be U, followed by again U. For T it will be A, then for G it will be C, for C it will be G, then for A it will be U, and then it will be G, then C, then 3 prime, which is the option number 1. Okay, so let me again repeat this thing. I know this thing might be little confusing, but if you read the options very carefully, and if you check this thing, you will understand. So the coding strand is not where the RNA it will make the difference. So the first we have to convert the coding strand to the non-coding strand, which is the template strand. So we have to just make it 5 prime, this to 3 prime, to the 3 prime, the complementary ones to 5 prime. So this is the template strand. Now the RNA strand, it will be complementary with this non-coding strand, which will be this one, and it will go to the 5 prime to 3 prime. So basically, if the coding strand is given, so remember the complementary RNA strand will be the same as the coding strand. So that is only the differences will be the thymine, it will be replaced by uracil. Okay, but if the question was like the non-coding strand was given, then you have to make it the complementary to answer it correctly. Okay, this was the problem number 9. Now I will move toward the next problem. So problem number 10 says the drug that inhibits the initiation step of the translation process is number A, ricin, number B, tetracycline, number C, streptomycin, and number D, cyclohexylamine. So which of the following drugs inhibit the initiation step of the translation process? Ricin, tetracycline, streptomycin, or cyclohexylamine? Try to answer this question. Which of the following drug inhibits the initiation step of the translation process? The drug that inhibits the initiation step of the translation process is ricin, tetracycline, streptomycin, or cyclohexylamine. let me check the chat box. I have one answer, it says number D. Let me check if number D is the correct answer or not. So the correct answer is, it is number C. Streptomycin is the drug that inhibits the initiation step of the translation process. So basically, what is the mechanism of action for the streptomycin? So streptomycin binds to the 30S ribosomal subunit. Streptomycin inhibits the S12 and 16S rRNA of 30S ribosomal subunit encoded by the genes RPSL and RRS. What is the wrong pairing of the codon anticodon reaction and misleading to the genetic code and blocks the translation. What is the mechanism of resistance? The resistance is due to the mutation of RPSL and RRS genes. So protein synthesis inhibition. So there are different drugs which inhibit the different steps of the protein synthesis. We have chloramphenicol, which binds with the 50S portion of and inhibits the formation of the peptide bond. We have the peptide protein synthesis side. We have the streptomycin. It changes the shape of the 30S subunit and causing to the code on the mRNA to be read incorrectly. And then we have tetracyclines which interfere with the attachment of the tRNA to mRNA ribosomal complex. So these drugs, they inhibit the protein synthesis, and this protein synthesis inhibition is a very good mechanism of action for the antibacterial drugs. Okay, so several antibacterial drugs, they acts like this. They so as you can see, this 50S portion and 30S portion of ribosomal subunit means it is a prokaryotic ribosome, means like a bacterial ribosome. So these drugs are acts as antibacterial agents which inhibit the protein synthesis in bacteria, and that's how they exert their antibacterial activity. So remember, streptomycin, it inhibits the initiation step by misreading the codon anticodon sequence, by thus blocking the translation, and there are tetracyclines and chloramphenicol also, which also participate in the protein synthesis inhibition. So now I will move toward the next problem. Problem number 11 says DNA polymerase 1 in E. coli or Escherichia coli has a specific function of number A, removes and replaces primers, number B, DNA elongation, number C, DNA repair, and number D, all of the above. So DNA polymerase, as I have told you already, that DNA polymerase is not a single enzyme, it is a group of enzymes, it is a family of enzymes. We have different DNA polymerases. So you have to identify in DNA polymerase 1 in E. coli has a specific function. What is the function? Number A, it removes and replaces primers. Number D, it has a function in DNA elongation. Number C, DNA repair, and number D, all of above. So let me check the chat box. Okay, I have two answers. I think I have two answers. It says number D and then number A also is given answer. Okay, let me check if number A is the correct one or number D is the correct one. So the correct answer is, it is number A. It removes and replaces the primer. So there are different DNA polymerases, as I have mentioned. The DNA polymerase is a group of enzyme. Okay, this is a family of enzymes. There are DNA polymerase 1, 2, 3, 4 and 5 also. So the characteristics of the DNA polymerases in E. coli. So first is the class of the enzymes like DNA polymerase 1, 2, 3, 4 and 5. Then first one is the 5' to 3' polymerase activity, which is yes in case of the DNA in case of all the 1 to 5 DNA polymerases. Then 3' to 5' exonuclease activity, this is yes for 1, 2 and 3, but no for 4 and 5. Then 5' to 3' exonuclease activity, only DNA polymerase 1 shows this kind of activity in E. coli, but not the others. So what are the functions? So DNA polymerase 1, it has a function in removing and replacing the primers. DNA polymerase 2 in function in DNA repair, restarts the replication after damaged DNA, halts the synthesis. Number 3, it elongates DNA. Number 4, DNA repair, and DNA polymerase 5 with DNA repair, translational and DNA synthesis. So these are the functions and different DNA polymerases in E. coli. Okay. So DNA polymerase 1, it has a specific function for removing and replacing the primers. Now I will go to the next problem. So problem number 12 says, which of the following is responsible for the initiation of RNA polymerase activity in bacterial strain? Your options are number A, initiation site, number B, promoter region, number C, sigma factor, and number D, rho factor. So which of the following is responsible for the initiation of RNA polymerase activity in bacterial strain? Initiation site, promoter region, sigma factor or rho factor? Which of the following is responsible for the initiation of the RNA polymerase activity in bacterial strain? Let me check the chat box. Okay, I have one answer. It says number C. Okay, let me check if number C is the correct answer or not. So the correct answer is yes, it is number C, sigma factor. So sigma factor is responsible for the initiation of RNA polymerase activity in bacterial strain. So what is a sigma factor? It binds the core enzyme to convert it to the hollow enzyme. It is encoded by RPOD gene. It has a critical role in promoter recognition but is not required for transcription elongation. It recognizes the correct promoter site by decreasing the affinity of the enzyme at the non-specific DNA sequences. The amount is only 30% to the amount of the enzyme. So this is the promoter engagement. So as you can see, there is a DNA 5 prime to 3 prime is this strand, and this strand is 3 prime to 5 prime. So this is the RNA polymerase. Okay, and here we have initiation site which has -10 element and -35 element. We will discuss it later again. So first is the initiation, okay, and then is the elongation, where there is a sigma factor. The release of the sigma factor and the growing RNA strand is the start point here, which is the responsible for the initiation, and the termination step, where there is the termination, so release of the RNAP, completed RNA strand, and the sigma factor, it rebounds to the RNA, it is the RNA polymerase to start another cycle of the translation. Okay, so remember, the sigma factor is the core enzyme to convert the RNA polymerase to the hollow enzyme, and is responsible for the initiation of RNA polymerase activity in the bacterial strand. Now I will go to the next problem. So problem number 13 says the reaction in the DNA replication catalyzed by DNA ligase involves number A, addition of a new nucleotides in the leading strand, number B, addition of the new nucleotides in the lagging strand, number C, base pairing of the template and newly formed DNA strand, and number D, formation of a phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next lagging strand. Which of the following is the correct reaction catalyzed by the DNA ligase? Again I will repeat, number A, addition of the new nucleotides in the leading strand, number B, addition of the new nucleotides in the lagging strand, option C, base pairing of the template and the newly formed DNA strand, and number D, formation of the phosphodiester bond between 3' OH of one Okazaki fragment and 5' phosphate of the next on the lagging strand. Identify the correct reaction. Let me check the chat box now. Okay, I have two answers. I think number D is the given answer. Let me check if number D is the correct one or not. So the correct answer is yes, it is number D, formation of the phosphodiester bond between 3' OH of one Okazaki fragment and the 5-phosphate of the next of the lagging strand. So DNA ligase, it seals the nicks between the Okazaki fragments, converting them into a continuous strand of a DNA, and covalently it closes the nicks of the double stranded DNA. So this is the basically the replication process for the DNA Okay, the first step, so here we have a double stranded DNA which is converted. So for the one is the 5 prime to 3 prime, which is known as the leading strand, and the 3 prime to 5 prime is known as the lagging strand. So first, the helicase is the enzyme that unwind the parental double helix. Okay, and then we have the single strand binding proteins that stabilize the unwound parental DNA. Then we have the DNA polymerase, which the leading strand is the 5 prime to 3 prime, and it in the leading strand what happens, the DNA synthesis is done by continuously in 5 prime to 3 prime by DNA polymerase. And then we have a lagging strand which goes from the 3 prime to 5 prime. Here the DNA in the lagging strand is synthesized discontinuously. Okay, the primase is synthesizes a short RNA primer which extended by DNA polymerase to form Okazaki fragment, and after RNA polymer is replaced by the DNA by another DNA polymerase, the DNA ligase joins the Okazaki fragment and from a growing strand. Okay, so DNA ligase, the overall direction of replication is 5 prime to 3 prime. In the leading strand it is done by continuously, but in the lagging strand it happens discontinuously, but the direction will be the same from 5 prime to 3 prime. So remember, there are so in the case of the particular DNA replication, there are so many enzymes which are important, like we have helicase, we have DNA polymerase, we have primase, we have the ligase, then we have the topoisomerases also are there, which is not shown here, which relaxes the tension between the when the DNA is unbound. So this is so the correct answer was number D. Okay, I will move toward the next problem. Problem number 14 says the recognition sequence of Pribnow box -10 is number A 3 prime TA AT 5 prime, number B 3 prime TT GA CA 5 prime, number C 5 prime TA TA AT 3 prime, and number D 5 prime TT GA CA 3 prime. So which is the correct recognition sequence of the Pribnow box -10? The recognition sequence of Pribnow box -10 is which one? What is the recognition sequence of Pribnow box -10? Let me check the chat box now. Okay, I have two answers. It says number C. Okay, let me check if number C is the correct answer or not. So the correct answer is yes, it is number C, 5 prime TA TA AT 3 prime. So Pribnow box lies 10 nucleosides from the transcription start point, which is also abbreviated as TSP, and a second was found 20 nucleosides away. Okay, so Pribnow box, it is minus 10, it is known as TAT, and it is from the 5 prime to 3 prime. Another one is the minus G, it is TTGACA. Okay, and so the diagram is of the promoter. This diagram is of the promoter region. The RNA polymerase does not copy the promoter sequence into mRNA, and we know this because the promoter is to the right of the transcription start point. The transcription start point is to right of the promoter. RNA new polymerase cannot copy promoter sequences. Minus consonance sequences are copied into DNA but not RNA. Promoter is copied but spliced into mRNA. So promoters are specific regions in DNA that are recognized by RNA polymerase. So there are start site, which is the first base to be copied to the RNA, and minus 10 sequence is the Pribnow box sequence contained within the all prokaryotic promoter region, and minus 35 sequence is the second recognition site. Sigma subunit recognizes this sequence and connects with it, and the RNA polymerase then comes into the contact with the Pribnow box. Okay, so the correct answer was number C. So now problem number 15, it says that the enzyme responsible for the bond formation between amino acid and the tRNA is number A, RNA polymerase, number B, amino acyl tRNA synthetase, number C, tryptophenyl tRNA synthesis, and number D, helicase. So which of the following enzyme is responsible for the bond formation between amino acid and tRNA? Your options are number A, RNA polymerase, number B, amino acyl tRNA synthetase, number C, tryptophenyl tRNA synthesis, and number D, helicase. Let me check the chat box now. Okay, I have different answers. I think it is number A and number B, or I think number B is the final answer given by a student. Let me check if it is the correct one or not. So the correct answer is yes, it is number B, amino acyl tRNA synthetase. This enzyme is responsible for the bond formation between the amino acid and tRNA. So tRNA, we know it has an anticodon sequence which binds with the codon of the mRNA, and it acts as a messenger. It acts as a carrier of the amino acid. So how it does it? So there we have the 3' position we have a free OH, and then there is a amino acid which attached to the 3' position. Okay, but which is the particular enzyme which is responsible for it? It is the amino acid tRNA synthetase. Okay, then this amino acid attach the amino acid to the 3' area, and it converts the ATP to AMP and PPI. So basically amino acid tRNA synthetases in tRNA amino acid charging. Okay, so we have this is one example, which is the tyrosyl tRNA synthesis, so which is for tyrosine, and we have AUA. This is the codon this is the codon for tyrosine, and then the tyrosine amino acid is here. So that's it how it binds. The covalent bond formation happens between the 3' position of the tRNA and the amino acid, and on the enzyme amino acid tRNA synthetase. Okay, so the first step is the tRNA and its cognate amino acid enter to the active site of specific synthetase. Using ATP, the synthetase catalyzes the covalent bonding between the amino acid and the tRNA, and then tRNA charged with the amino acid is released from the synthetase enzyme. Okay, so remember, amino acid tRNA synthetase is the enzyme, and it is particular for a particular amino acid. I will go to the next problem next. So problem number 16 says the colonies of E. coli grown in the 15NH4Cl medium are transferred to a medium having 14NH4Cl as a nitrogen source. The DNA isolated after 2 cycles of the growth was subjected to density gradient centrifugation. What is the following figures show the correct presentation? A, B or C or D? Again I will repeat, colonies of E. coli grown in the 15NH4Cl medium are transferred to a medium having 14NH4Cl as nitrogen source. The DNA isolated after 2 cycle of growth was subjected to density gradient centrifugation. Which of the following figures show the correct presentation? A, B, C or D? Let me check the chat box now. Okay, I have one answer. It says it is number D. So I have two answers saying it number D. Okay, let me check if number D is the correct answer or not. Yes, number D is the correct answer. So this is known as Michelson-Sterl experiment. So this is the DNA that it is a hypothesis is the DNA replicates semi-conservatively. So first we have a vessel where the bacteria grows in 15 nitrogen or heavy nitrogen medium, and then after some time, so this is the sample. If we sample it at 0 minutes, so we will have only one mark of the the 15 and 15 heavy DNA. Then if we transfer some bacteria to a lighter medium, which is the 14N medium, and if we do it up to the 2 cycles. So first cycle is take the samples after 0 minute, 20 minutes after 1 round, and 40 minutes after the 2 round. So as after the 40 minutes, so what you will see, so there is a 2 mark here. So what does it denotes? That after 2 generation, half of the DNA was the intermediate and half of the light only, and there is a no heavy DNA. So at the first, at the 0 minute, we have only the parental heavy DNA. For the first generation, we have a mixture of 14N and 15N intermediate. Okay, and then at the second strand and the last one we have two strands which have only the light strands. Okay, so this experiment, this pattern can only have been observed if each DNA molecule contains a template strand from the parental DNA, thus DNA replication is semi-conservative. Okay. So now I will move toward the next problem. Problem number 17 says which of the following statements regarding RNA is false. Your options are: number A, rRNA acts as a catalyst to make the peptide bond. Option B, the amino acid sequence of the proteins depend on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes the mRNA. And number D, RNA polymerase 1 synthesizes the rRNA. So which of the following statement regarding RNA is false? Number A, rRNA acts as a catalyst to make a peptide bond. Number B, the amino acid sequence of proteins depend on the base sequence on mRNA. Number C, RNA polymerase 3 synthesizes the mRNA. And number D, RNA polymerase 1 synthesizes the rRNA. Which of the following statements regarding RNA is false? Again, I will give you 1 to 2 minutes. Let me know in the chat box A, B, C or D which of the following is the correct answer. Okay, let me check the chat box if there are any answers. Okay, so I have one answer which says it is number C. Let me check if it is the correct or not. So correct answer is number C, RNA polymerase 3 synthesize mRNA. This statement is false. So RNA polymerase also, like DNA polymerase, has different kind. So RNA polymerase 1, the RNA polymerase that only transcribes the ribosomal RNA but not 5S rRNA. RNA polymerase 2, the central enzyme that catalyzes DNA directed mRNA synthesis during the transcription of protein coding genes. RNA polymerase 3, so basically it is the RNA polymerase that transcribes small untranslated RNAs such as tRNAs. RNA polymerase 1 transcribes rRNA genes. RNA polymerase 2 transcribes mRNA, miRNA, snRNA and snORNA genes. RNA polymerase 3 transcribes the tRNA and 5S rRNA genes. RNA polymerase 1 resides in nucleus and RNA polymerase 2 resides in nucleoplasm and RNA polymerase 3 resides in the nucleoplasm. RNA polymerase 1 transcribes the pre-rRNA at 200 copies. Polymerase 2 transcribes the around 25,000 genes, and RNA polymerase 3 transcribes 30 to 50 genes at variable copy numbers. RNA polymerase 1 contains the same two non-identical alpha-like subunits. RNA polymerase 2 has two copies of the different alpha-like subunit, and RNA polymerase 3 contains the same two non-identical alpha-like subunits. RNA polymerase 1 is insensitive to alpha amanitin RNA polymerase 2 sensitive to 1 microgram per ml alpha amanitin, and RNA polymerase 3 sensitive to 10 microgram per ml alpha amanitin. Okay. So problem answer number C was the correct answer. I will go to the next problem. Problem number 18 says the enzyme responsible for removing the introns and joining the exons together are called number A lysosomes, number B ligase, number C spliceosome, number D helicase. So which of the following enzyme is responsible for removing the introns and joining the exons together? Lysosome, ligase, spliceosome or helicase? Try to identify the correct option. The enzyme that is responsible for removing the introns and joining the exons together. Let me check the chat box now. Okay, I have one answer. It says it's number B. Okay, let me check if number B is the correct answer or not. So the correct answer is number C, spliceosome. Okay, spliceosome is the enzyme which is responsible for removing the introns and joining the exons together. Spliceosome is a large and complex molecule formed of RNAs and proteins that regulate the process of RNA splicing. The spliceosome is composed of 5 small nuclear RNAs and about 80 protein molecules. The combination of RNAs with these proteins result in the formation of an RNA protein complex termed as a small nuclear ribonucleoprotein or SNRNPs, and these are most commonly confined within the nucleus, where they remain associated with the immature pre-RNA transcripts. So there is a DNA. So DNA, there are some parts of the DNA which are not required for the mRNA. Okay, so these parts which are not required are known as intron, and the parts which are necessary are called the exons. And spliceosome, what does it do? It removes the introns, joins the exons to form a mature RNA or mature mRNA. Okay, so remember, spliceosome is the enzyme which is responsible for removing the introns and joining the exons together. I will go to the next problem. Problem number 18 says which of the following amino acids is involved in the action of human topoisomerase 2? Your options are number A tyrosine, number B tryptophan, number C serine, and number D proline. So which of the following amino acid is involved in the action of human topoisomerase 2? Tyrosine, tryptophan, serine or proline? Okay, let me check the chat box if there are any answers. Okay, number A is the given answer. Let me check if number A is the correct one or not. So the correct answer is very good. It is number A, tyrosine. So tyrosine is the amino acid which is involved in the action of human topoisomerase 2. So topoisomerase 2 is a mammalian enzyme that is crucial for effective replication of DNA. The enzyme binds to the parts of DNA where two regions of the double helix are near proximity. The enzyme binds to one of these DNA double helices, and tyrosine residues are used to nick both strands of DNA. This results in temporary covalent bond between the enzyme and resulting 5' end of the each strand, thus stabilizing the DNA. The strands are now pulled in the opposite direction to form a gap between which the intact DNA region can be passed, and the enzyme then reseals the strands and departs. So method by which DNA topoisomerase 2 catalyzes the crossover DNA strand, and as you can see, there is a two crossover DNA strands are here. So there is a topoisomerase with the tyrosine amino acid, and the tyrosine it binds with the one strand and another, and it when the tyrosine is binding, it separates the two interlocking DNA strand, thus relieving the tension. Okay, so how does it do? The mechanism by which topoisomerase 2 splits the DNA chain. So we have the tyrosine attached with the topoisomerase 2. The tyrosine have a hydroxyl group, and this hydroxyl group attacks to the phosphodiester bond, and that's how it makes and that's how it cleaves the particular DNA chain. Okay. So problem number 19, it says which of the following is not an inhibitor of DNA polymerase? Your options are number A, cytarabine, number B, gamecitabine, number C, fludarabine, and number D, darunavir. So which of the following is not an inhibitor of DNA polymerase? Cytarabine, gamecitabine, fludarabine, or darunavir? Okay, let me check the chat box. Okay, I have one answer. It says number D. Let me check if number D is the correct answer or not. Yes, number D is the correct answer. Darunavir is not an inhibitor of DNA polymerase. So DNA polymerases catalyze the synthesis of DNA during the using the four deoxyribonucleotide building blocks like DATP, DGTP, GCTP, and GTTP. The anti-cancer drug cytarabine is an analog of 2'-deoxycytosine and acts as a prodrug. It is phosphorylated in the cells to the corresponding triphosphate ARA CTP, which acts as a competitive inhibitor. In addition, it can act as a substrate for DNA polymerase and become incorporated into the growing DNA chain, and this can lead to a chain termination or prevent replication of the modified DNA. All of these effects result in the inhibition of DNA synthesis and repair. Gamecitabine is an analog of cytarabine and has a fluoride side effect. The purine analog fludarabine is also metabolized as a triphosphate and has the same mechanism of action of cytarabine. It too inhibits the transcription and can also be incorporated into the RNA. So remember, cytarabine, gamecitabine, and fludarabine, these three are the inhibitors of DNA polymerase, but darunavir is not. It is a anti-HIV drug. Okay, it is a it is an antiviral drug, not a DNA polymerase inhibitor. So now last question of today's session says that which of the following enzyme is a helix breaker? Your options are number A, DNA polymerase, number B, DNA ligase, number C, helicase, and number D, lysozyme. So which of the following enzyme is a helix breaker? DNA polymerase, DNA ligase, helicase or lysozyme? Okay, let me check the chat box. I have one answer, it says it is number C. Let me check if number C is the correct or not. Yes, number C is the correct one. Helicase is the enzyme which is known as the helix breaker. So helicase uses an energy from the ATP to break the hydrogen bonds holding the base pairs together. This allows the two parental strands of DNA to begin unwinding and forms two replication forks. Each strand of parental DNA has its own helicase. In humans, two inherited diseases, Werner's syndrome and Bloom syndrome, results from the helicase defects. E. coli contain at least 6 different helicases, some involved in DNA repair and others are conjugation. Principal helicase in the DNA replication is DNAB. So helicases are often used to separate the strands of DNA double helix and or a self-phenyl RNA molecule using energy from ATP hydrolysis, a process characterized by the breaking of hydrogen bonds between the annihilated nucleotide bases. They also function to remove the nucleic acid associated proteins and catalyze the homologous DNA recombination. Okay. So this was the last question of today's session. So today we discussed about the central dogma, we discussed about the many things related to the DNA replication, protein synthesis, and also the we discussed about the enzymes which are responsible for them. Now I will share my email ID in the chat box. So this is my email ID. So if you have any doubt, any query regarding today's class and in general regarding the week 6 course content, you can let me know through email. I will close the session for today. If you have any doubt and any query, please feel free to let me know through the email. This presentation and class recording I will share to you you can check it later tonight or tomorrow in the SWAYAM portal. Okay. So I will close the session for today. Thank you all for joining, and we will meet in the next Monday from the 6 p.m. to 8 p.m. Thank you all. I will close the session for today. Thank you. I am closing the session.